Hello.
Surprise!
Mary Claire says hi. And
I
she's
do say
fixing
hi.
my hair because it's sticking up, so you have to ignore what's going on over here with my hair
Yes.
while you take notes.
Okay, well, I have to leave. So good luck, you person. Awesome. Well, I actually do have to leave. Bye.
Bye. Okay, so let's discuss when we have biochemical evidence and the importance of that, the importance of DNA when we are taking a look at common ancestry. So if you look at D.5.2, we want to explain the biochemical evidence provided by the universality of DNA, as well as protein structures within the common ancestry of living organisms. So we are looking at DNA as being universal, and we can say that it's universal because we know that all organisms use DNA as genetic material. Anything that's a living organism has DNA. And we can see that the genetic code is universal because if we take a sequence of a gene and put it within a different organism, the same proteins are going to be expressed, which therefore can tell us that the genetic code is universal. Secondly, we can take a look at our biochemicals. We can look at amino acids, and we know that amino acids make up proteins that are within our body. And there are about 20, maybe 22, 23 amino acids that are used to make every single protein that we have. And the combination of that can make different types of proteins. We know that most amino acids can exist in a left or a right hand form. And so we have on the screen left and right. You've got two different forms of amino acids, and this is an organic chemistry discussion, but the way that any sort of compound can come together, organic type compounds, is that there's a left and right formation. It just simply means that something is mirror image. And I don't want you to get too hung up on this, but there is a difference because they're mirror images, and pretty much any living thing uses this particular formation right here. It's going to use the left-hand amino acids. And we see this through the panspermia hypothesis because what they're seeing is that there were the amino acids contained on meteorites are in the majority left-handed. So if this particular theory was true for evolution, then that would hold true that majority of our amino acids are in that left-handed stage. So let's look at specific molecules and variations within very specific molecules that may show us differences in our lineage. And so we have a very specific protein called cytochrome C. We see it involved within the electron transport chain, and it has about 100 to 104 amino acids, and this is it's found within plants, animals, and unicellular organisms. So it is essentially located everywhere. And it's pretty complex, so the thought is if it is very complex, it must have evolved independently. When you think about the complexity of mitochondria and chloroplast, we have determined that it has evolved independently, and so this is kind of similar. And with that being said, there must be a common ancestor because it has evolved and branched off independently. So this protein cytochrome C is not identical in every single species because there is a single point mutation within the DNA. And that single point mutation can lead it to be a different amino acid that is making up this particular protein. So humans and chimps have that cytochrome C molecule, and rhesus monkeys, which we see at the right, they share all of the amino acids within that chunk of amino acids, except the 66th amino acid is actually different. Okay, in rhesus monkeys, it's isoleucine, but within our chimps and humans, it's threonine. Therefore, because there is that difference in that single point mutation, this suggests that humans and chimps are more closely related to each other than to rhesus monkeys. And I know this seems like a very small, subtle change, but it's enough to change the phylogeny that maybe had existed prior to having the tools to study amino acids within the DNA. So, if we look at these base sequences, we can assume that 1, 2, 3, and 4 are all different animals, and we want to determine which of the species are most closely related. And so we can compare all the amino acids. For example, we'll compare it to species number 1, and we can determine which are closely related. So I've done that for you so that we don't have to do the labor. We will practice this within some labs coming up. But right here, this circle is representing the differences when we're comparing it to species 1. So species 1 is our kind of standard, the species we want to compare to, and they're circling the differences that we see. So if you note, species 3 and 4, they only have one base pair difference. So we can say that species 3 and 4 are most closely related to species number 1. While we look at 1 and 3, they have the most differences of about 3 base pairs. Okay, so right here, 1, 2, 3. So when we are looking at them as a whole, this particular species, number 3, is most distantly related. And this is by taking out this group of amino acids, comparing it to other animals, and this is the same exact area of amino acids. It's the same gene that we are looking at. Now moving on, we want to discuss how biochemical variations can actually be used as kind of a timeline. Not necessarily like a clock on the wall, but we can use it to have a better estimate, a better hypothesis of evolution and phylogeny. So an evolutionary clock is going to involve calculating the time since species have diverged by comparing the number of differences within the DNA. So we're looking at differences within DNA. It could be looking at protein sequences, depending on what animals we're looking at, and we can determine the divergence. So a polypeptide sequence may accumulate at a steady rate or gradually over time, and so we can look at it as some sort of marker. And we're going to look at homologous traits, homologous molecules of related species, and then we can count the base pairs that do not match, very similarly to what we have done in the previous lesson. And so what we can do is assume that the changes that are occurring within this phylogenetic tree are happening at the same rate. And this isn't always the case. You would have to be aware that this is occurring. So if we look at species A, it may have 5 differences between species B. Okay? And then from A to C, the differences may be 10 base pairs. So right here, we have 5 base pairs. Right here, from A to C, it could be 10 base pairs. So the assumption is that between A and C, the split happened twice as long ago than compared to A and B. So we are looking at a proportion or some sort of estimation. We are not looking at absolute time. If we want an absolute time, we would need to look at carbon dating and get specifics within the strata levels of the Earth at that time.